Entonces me está diciendo, ¿dónde está tu link que dijiste que ibas a mandar a las 12?
Luego está pidiendo ayuda. Está diciendo de seguro.
¿Sí ya se ve?
Así está el departamento de farmacobiología. Se ve así bien.
Sí, ahorita quiero ver si se escucha. Sube el volumen, ¿no?
¿Se escucha?
¿Sí? ¿Lo que dijimos se escuchó?
Con el micrófono se debe escuchar más, supongo. Aunque hablen así de bajito, se escucha.
¿Ah? Sí.
No, yo creo que no.
Bueno, ya le pedí permiso al profesor Traub de presentarlo en español. La charla, por supuesto, va a ser en inglés, ¿no? Bueno, el profesor Roger Traub es uno de los pioneros en el modelaje matemático de neuronas, de ensambles neuronales, del sistema nervioso central. Es uno de los pioneros en introducir y darle una importancia muy grande a las gap junctions, a las sinapsis eléctricas, que explican varias cosas que él predijo con sus modelos matemáticos. Y el doctor Traub primero estudió en Princeton la carrera de matemáticas y después estudió en Pensilvania medicina. Después hizo una residencia en neurología, entonces es matemático, neurólogo. Es actualmente investigador del Instituto IBM de Watson en Nueva York y su tema de interés y en el cual está involucrado es, además del desarrollo de estos modelos, porque él primero hizo un modelo de una neurona y de varias neuronas comunicándose. Además de eso, pues es la epilepsia, por supuesto, uno de los eventos electrofisiológicos más notables con los que él ha trabajado bastante. Y les puedo decir que ha recibido innumerables preseas y reconocimientos, entre ellos pues, uno muy grande de la Sociedad Americana de Epilepsia, el premio Humboldt. Es mi hermano mayor en ese sentido, yo fui becario Humboldt, y en la Fundación Humboldt en Alemania se tienen 3 tipos de premios. Uno es el Junior para jóvenes postdocs, otro es el premio de investigación, que fue el que me dieron hace 20 y tantos años, y otro es el premio Humboldt, que es para personalidades como la del doctor Traub, y tiene varios libros, uno de los cuales lo pueden encontrar muy fácilmente, fue publicado en el 2010, que se llama Cortical Oscillations in Heart and Disease. Entonces, now I switch to English, first to welcome you in Mexico, and thank you for giving us this talk.
Thank you very much. Rafa was kind enough to invite me here, and Rafa has done and is doing wonderful things with gap junctions, particularly with mixed synapses, and we have collaborated on this in the past. But what I'm going to talk about today is actually something a bit different than what Rafa does. It does involve gap junctions, and it came out of some accidental findings in England in 1998 and in Germany at that time. So the title is Gap Junctions and Very Fast Oscillations in the Brain, of course. Let's see if I can get this to work. So these are the questions I want to address. And if I'm speaking too fast or too obscurely, please interrupt, because I don't have. So the first question is, what is the evidence, experimental evidence, for gap junctions between pyramidal neurons? And the context of this is other types of gap junctions are very well known and accepted in the inferior olive, or between many types of interneurons in the cortex and hippocampus, but this is a much more controversial question. Question 2 is, it sort of assumes gap junctions can account for very fast brain oscillations, ripples in other words, which I'll try to convince you of, and explain the mechanism by which that's possible. And the reason this is a question is people think of gap junctions as producing synchronization between cells, and if that's the case, it's not clear how you would get a collective oscillation out of a network of coupled neurons. Assuming you can do it if the neurons are intrinsic oscillators, but if they're not intrinsic oscillators, it's not obvious how to do that. But it's simple once you see it. And if I have time, I'll talk about what are the implications for epilepsy, which I'm especially interested in, and normal brain function. And I'm sure I won't have time for this. So, some background on ripples. So, physiological sharp waves were described in the hippocampus probably in the 1970s. Certainly, there were papers about them in the 1980s, and their potential looked like something like this. This was in vivo in a rat, and they're quite enormous. And they would occur during slow wave sleep or during wake immobility in rodents. So this is a very famous paper that was published in Science in 1992 by Yuri Bujaki and colleagues. And what it is about is that when you have a sharp wave like this, which lasts about 100 milliseconds, superimposed on it is this very fast, in this case about 200 hertz, field oscillation. So here's a broadband recording, here's filtered, and this is 100 to 400 hertz. This is roughly 200 hertz. So this paper has been quoted countless times, And the study of sharp wave ripples is now an industry, especially because it is believed that this kind of event is somehow related to memory consolidation. So I don't want to talk about that. What I want to talk about is what is the cellular mechanism by which an event like this can come about. So one technique that helps to understand this question is this kind of phenomenon has been replicated in brain slices. So this is another famous paper by Nikolaus Meyer and Dietmar Schmitz and Andreas Dragun. And they found that if you take a slice of hippocampus, especially ventral hippocampus, you can observe events like this. This is a sharp wave, this part here, kind of like that. And then there's a superimposed ripple oscillation. So these occur spontaneously in a slice preparation without drugs. And because you can observe these in vitro, it's possible to do all kinds of pharmacological and electrophysiological measurements. That doesn't mean that the mechanism is worked out. It's just something that can potentially be worked out. Now I want to switch a little bit. So people think about gap junctions as resistors, which they are. Electrically, they're like resistors. And if you pretend that neurons are linear devices, which of course they're not, then a resistor between two quote-unquote linear neurons will act like a low-pass filter in an RC, a resistor capacitor circuit. And the physics of that will be it will produce DC coupling or low frequency coupling. But there's another way to look at what gap junctions do if you take into account the extreme nonlinearities of neural membranes, particularly when you're dealing with axons, which have a very high density of sodium channels and are designed to be excitable because they have to transmit action potentials. So one of the earliest papers, this is from 1959, that demonstrated directly electrical coupling between neurons showed that this very nonlinear type of behavior. So this is from a crayfish nervous system. And First-Pan and Potter were able to get 2 electrodes into this giant axon and into this large but not giant axon, and showed that they were electrically coupled in such a way that an action potential in one axon, this one, would induce with probability 1 or nearly 1 an action potential in the other axon. So the gap junction here is not acting like a low-pass filter. What it's doing is it's acting like a pulse transducer. It allows spikes to go from one axon to another axon. So this is key to how one might think about gap junctions between axons in mammalian nervous systems, assuming that those gap junctions actually existed. They might behave like this. So here, that was an invertebrate. The notion that there was electrical coupling between axons in vertebrates is actually also rather old. I mean, 1967 is probably old to most of the people here, not to me. But anyway, this is an electron micrograph from Mike Bennett and colleagues, one of the pioneers in gap junction studies, and it's from a fish. And this is one axon, this A is another axon, and the arrow points to what appears to be ultrastructurally a gap junction between them. So very little was demonstrated about the physiology of this coupling, but the morphology was shown. This again was a long time ago. So now I want to show some pictures of some of the people involved in this. This guy I think you all know. This is Andreas Dragun, who is in Heidelberg and is now unfortunately for him, the dean of the medical school in Heidelberg. This is Dietmar Schmitz in Berlin. This is Hanna Monnier, Heidelberg. I showed this picture to a colleague at IBM, and so they don't make them like that anymore. This is Eberhard Buhl, who died tragically 15 years ago in his 40s, and that's Miles Whittington, who is now in York in England. So here is the basic experiment, which is the foundation of this lecture. This was an accidental finding of Andreas Dragoon in the late 1990s when he was visiting this guy, John Jeffries, in Birmingham in the United Kingdom. And Andreas was in the lab to study something else, tetanus toxin, and he had hippocampal slice, and he was putting his electrode in field or recordings, and he noticed spontaneously ripples like this. So this was very odd, because in vivo, when you have a ripple, it's superimposed on this big potential. But what Andreas was seeing was the ripple without the sharp wave. And these would occur just by themselves. And so there are a number of questions that this observation raised. One is this a single unit? It's not. And what is the mechanism by which this happens? So many cells are somehow cooperating and producing this high frequency oscillation, but in a somewhat unusual way because you don't have this underlying wave. So one of the basic experiments Andreas did was he bathed his slice in a nominally calcium-free medium, a low calcium medium. He showed that chemical synaptic transmission was blocked. And when you do that, instead of abolishing these ripples, you get more of them. So this is produced, this is a collective phenomenon produced by interactions between cells without chemical synapses. So what could it be? So how can cells influence one another? Well, one way that they might influence each other is something that Jeffries was interested in, was called field effects, in which currents flow across middle membranes, and they flow in the extracellular medium and make loops and perturb membrane potentials. So this is a kind of called field coupling. So this seems to be impossible, and the reason is that the amplitude of these potentials is in the tens of microvolts, so that the perturbations of the neural membranes that would be produced by this collective phenomenon are of the order of the thermal fluctuations of the channels, and would be very unlikely to produce behavior involving many neurons synchronously. So we thought that was not possible. So what the other possibility was, it depended on gap junctions. And gap junctions between pyramidal neurons were suspected at this time. This is late 1990s, because earlier people like Ed Dudek and others had shown that there was dye-coupling between pyramidal cells. So if you inject a fluorescent dye into one cell, then it passes into other cells, and you see two or more cells kind of lighting up together. So their existence was suspected by some people and vigorously denied by other people like Eric Kandel, who still denies them as far as I know. So in any case, Andreas tried pharmacology using drugs that block gap junctions. Unfortunately, none of them are clean. They all have side effects, but the side effects of the different drugs are different from each other. So it turns out that a whole slew of different blockers all suppress the ripples. So this is halothane, which is a general anesthetic, was one, and they use carboxolone and Optimal. So the conclusion from this kind of experiment was you can get ripples, They're produced by electrical coupling somehow. And you don't need synapses, chemical synapses. So, how could this happen? Now, the prevailing idea at that time, and even now in most circles, is that ripples are produced by populations of interneurons, of GABAergic cells. And I think that this is incorrect. I'll try to convince you of that. But that was the prevailing idea. So Andreas recorded from a number of interneurons, or GABAergic cells in the slice during these ripples, and what he found was none of them paid any attention to this event. They simply were indifferent to the whole thing. So they were not generating the ripples, at least in these conditions. Now it's hard to do patch recordings of cells in low calcium media. It'd be impossible for me because I don't do these experiments, but even for experts it's hard to do. But Andreas was able to get one cell, and he showed it was behaving like a pyramidal cell with an adapting train of action potentials in response to a current pulse, and that cell wasn't ignoring the ripples, it was actually participating in the ripples, and it was doing it in a very unusual way. So, first of all, this tells you the ripple is collective because the amplitude of these perturbations, it's all about the same, but if you look at this neuron, sometimes it has a little action potential or a spikelet in phase with this, and sometimes it's a full action potential. So that tells you this cannot be recording this unit. I mean, it's recording in part this unit, but it's not identical to this unit because of this fluctuation between that and that. So then that raised the question, where are these spikelets coming from? And what is this telling us about the mechanism? So I then did some simulations of pairs of model pyramidal cells. And I won't go into details, but I put a simulated gap junction between the dendrites of two of these cells, and I made one of them burst with current pulses. And if you look at what happens in the other cell, you get sort of low frequency DC-like coupling. So all this stuff gets turned into this slow wave. On the other hand, just for the hell of it, I put a gap junction between axons, and now if one cell is doing this kind of thing, spikelets like this, which look very much like that, then that's what the other cell does. And the reason was that if you have a gap junction between two axons and spikes can cross from one to the other, like in a crayfish, then both axons will tend to fire together, and then the spikes in the axon, as they propagate backwards, can then block and produce a spikelet, or not block and produce a full antidromic action potential. So if we assume this, at least in two cells, then we get potentials that look like what was measured experimentally. It doesn't tell us how a large population of cells will do this kind of thing, but at least it looks right to a two-cell level. So, unfortunately, this paradigm has only been replicated once, as far as I know, and people, sort of low calcium media are hard to deal with in that preparation, but the basic idea has been replicated in other ways. Again, this is from this sort of the usual suspects. This is Volker Nimmerich, Niklaus Meyer, Dietmar Schmitz, and Andreas Traub. So they showed that they could observe spontaneous sharp wave ripples in a slice, and they could see them with this drug, which blocks GABA A receptors. So again, you do not need synaptic inhibition in order to get this phenomenon. So this is not in low calcium. This is with synaptic transmission present, but synaptic inhibition has been removed. So this is a little pulse from spritzing some potassium chloride and you get a nice ripple here. So now I want to sort of give an outline of how ripples really occur, at least how I think they really occur. Oh, that's Andrea Dede, who was a postdoc of mine in the past. So this is the model that we presented, which puts together the Dragoon findings on the calcium and some other data on, from Andreas Dragoon's lab, and into a consistent picture which makes very precise experimental predictions, which I will sort of lead to and show you that are verified. So to remind you, this is the standard notion of how ripples come about is from a population of interneurons that inhibit one another or are electrically coupled or something. And this idea didn't come out of nowhere. It came out of the in vivo observation that during a ripple, you have this fast oscillation, and fast spiking interneurons follow the oscillation very precisely. And if you record from a pyramidal cell during a ripple in vivo, you see 200 hertz IPSPs. So from those observations, it's quite logical to guess that this is what's producing the ripple. But there's another way to look at it. Let us suppose, so this sort of is the pyramidal cell population, which I've arbitrarily divided up into the cell bodies and the dendrites, as sort of one entity, and the group of axons of these cells is another entity, and the axons are coupled by gap junctions, which I gave some preliminary data supporting. And let's suppose that this is what generates the ripple, and not this. Now suppose this is what's generating the ripple. Then the output of this circuit, of this collection of axons, will produce ripple frequency synaptic excitation of the interneurons, and they will follow the ripple. So it explains that observation. And then if these cells are following the ripple, then they will produce synaptic potentials here, which also follow the ripple. So this is as consistent with the data, this idea, as this idea is. But this idea makes some other predictions. And one is that if this system is what's generating the ripple, then when you see action potentials here, they're antidromic. They're coming backwards from the axon. That's a very specific prediction. And not at all an intuitive one. And another prediction is, if this is what's generating the ripple, then you will see ripple frequency excitatory synaptic potentials here, as well as inhibitory synaptic potentials. So both of those observations are true. So I'll try to now kind of build up to that point. This idea is so entrenched that it's kind of almost impossible to dislodge it, but I'll make an effort with you guys. Assuming I can get rid of this. And get rid of this. So here is some evidence that pyramidal cells are actually electrically coupled. This is from pair recordings. This is from the lab of Alex Thompson in London. And she recorded, or her lab recorded, pairs of pyramidal cells in the hippocampus. And they also did it in cortex, but this is hippocampus. So this is the crayfish revisited, this one. So here there's a current pulse, depolarizes this neuron, it fires, and then this one fires. Just like a crayfish. Here there's a variation. This cell was induced to fire, and now you get spikelets. See this scale bar is 5 millivolts. But these are spikelets. They're like miniature action potentials. So this is direct physiological evidence from a pair of cells that there is electrical coupling between them. So, I don't know if I can quote your pair data or not. It's okay. So Rafa has shown something like this. It's even more impressive between granule cells and CO3 cells. But you can talk about that. So this is pairs of cells in layer 5 in the neocortex. So you're seeing the cells labeled here, and these investigators recorded from these pairs of cells, and they induced, again, a burst of spikes in one, and in the other cell, they would get spikes and spikelets, following these one-to-one. And they also noticed that this is light microscopy, that the axons of these cells kind of kiss each other. They're these very close appositions. That doesn't prove that there's a gap junction there, but it suggests that there might be. So here is more kind of light microscopic morphological evidence for coupling between pyramidal cell axons. So this was from a paper by Dietmar Schmitz et al. In 2001, and these experiments were done by Sebastian Schuchmann, who's a child neurologist. And what he did was, this is a fluorescent dye being injected into a CA1 pyramidal neuron, which is lighting up, and he was able to do these confocal imaging across a piece of tissue like this, and in 3 dimensions, up and down, and to do this repeatedly over time, over roughly 30 minutes. So by doing this over 30 minutes, he was able to follow the course of the dye from one cell to a second cell. And in 5 cases, he had enough resolution that he could see the dye go from this process to this process, and these processes, light microscopically, are axons. And you know that because they don't have spines, and because they have collaterals like this that come off at right angles. So this is morphologically an axon. This is a dendrite with the spines on it. Now I'm going to jump ahead. One source of controversy was the claim ripples depend on gap junctions between axons, and the most common gap junction protein in the brain is connexin 36. And it was noticed that in the early 2000s, Hannah Monnier developed a connexin 36 knockout mouse, and showed that interneurons, which are normally coupled by connexin 36, became uncoupled. But in the knockout, ripples still occur. So that led some people to say, oh, connexin 36 is gone, therefore these cells cannot be coupled by gap junctions, ignoring the fact that in other parts of the body, and especially in the retina, which is really part of the brain, there are many other connexins, including connexin 45. So Jim Nagy, or Nut, if you're Hungarian, he's a specialist in developing antibodies to connexin proteins, and he has developed antibodies to 36 and 45 and many others. And he came up with this remarkable set of images, which unfortunately is still not published, but he's given me permission to show it. And what you see here is this is double labeling. So he has antibodies to anchorin G, which is a protein that you find in the initial segments of axons. So this stuff that looks like seaweed, these are actually bundles of proximal axons of CA1 pyramidal neurons. And then he also labeled this tissue with antibodies to connexin 45, and when you superimpose them, you see that higher power is this. So these are the axons, and then this is connexin 45 labeling. So, and then sometimes, when the axons cross, you see a blob like that. So this is very suggestive that there's a gap junction here, but does not prove that there's a gap junction there. And in fact, one of the bad things about this is there's so much connexin 45 that it could be that what the cell is doing is it's incorporating blobs of this protein in the membrane, and they float around, and then every now and then, when there's an apposition, they're able to actually make a functional gap junction. But of course, that's hard to prove without freeze fraction. Now, another indirect bit of evidence for connexin 45 being involved is from this study from Janelia Research in Virginia. So this is Nelson Spruceman and colleagues, and they did a RNA sequence database for principal cells in the hippocampus. And you can find the details. You have to go to the website. So if you go to this paper in eLife, there's a link to the website. And what this shows is that the protein, this is the gene, GJC1, is a gene for connexin 45. So this mRNA is very highly expressed in pyramidal neurons, at least CA1 pyramidal neurons. And for connexin 36, it's at very low levels in CA1, but I take it that's not true in CA3. Okay. So now let me return to spikelets. This is an area where people have strong opinions. I'm not sure why, but Richard Miles, who's just retired from Salpêtrière in Paris, he and I have known each other a long time, and we wrote a book together. He has seen spikelets since very early in his career, and he never takes them seriously. He's convinced, for whatever reason, that they are some kind of an artifact and that the cell is sick. So that's Richard. So that's one opinion from somebody who I respect a great deal. But there are other people who wonder about this, that the spikelets are not artifacts of sick cells in brain slices where axons and dendrites have been cut. And the reason for saying that is that spikelets have been observed in vivo, and for a long time ago. So this is a classic series of papers from 1961 by Alden Spencer and Eric Handel. And what you see here, this is a anesthetized cat, is a spontaneous occurrence of spikelets. And here's another one. And in this classic study, they showed this potential involves some kind of active membrane process because it's decaying very quickly, unlike what you would expect just from the RC properties of the membrane. This is classic work. So they occur spontaneously in vivo and are presumably not an artifact of a slice preparation. Here's another example from Spencer and Kendall. And what is interesting about this is this spike looks antidromic. So it looks as though it's coming from the axon. And the reason for thinking that is that it's arising from a hyperpolarization and that there's an inflection in the rising phase. These are characteristics of antidromic spikes. And so now Dietmar Schmitz then undertook to test the idea that axons were coupled with a series of very elegant electrophysiological experiments. So this is the same paper. I showed this dicoupling from this paper. I just want to review some of the electrophysiology. So we're in a slice. We're in low calcium media. For most of the experiments, chemical synapses have been blocked, and Dietmar is patching onto the soma of a neuron, pyramidal neuron, and he's found if he stimulates nearby, he'll have a classic antidromic spike coming from the axon. But he noticed that if he moves the stimulating electrode sort of over here and stimulates, now he sees spikes that look like this. And if he reduced the amplitude of the stimulus, he gets spikelets. And he showed that if he stimulates repetitively at very high frequency, the spikelets will follow the stimulus one-to-one without any failures, up to 400 hertz. This is 200 hertz. Now, why do I go into that? The reason is that in the 1960s, when Spencer and Kendall saw this phenomenon, they realized that these were action potentials coming from someplace, and they postulated that they're coming from the dendrites. I don't have a picture here, but their proposal was that you have the pyramidal neuron, so that's the cell body, basal dendrites, apical dendrites, that somewhere up in the apical dendrites there was an action potential, and that it would propagate toward the soma, but block, and then produce that. It turns out that much later it was shown that dendrites, if they're stimulated strongly enough, at least in many kinds of pyramidal cells, really can make action potentials. But when they do, what you record at the soma doesn't look like this. It looks like a low-pass filtered version of this. On the other hand, if the spike is coming from the axon, as I'll show, then you would expect to see potentials like this. So Schmitz et al. Did a series of experiments. I won't go into them all, but this is the idea that this is the hypothesis that there's a gap junction there, or approximately there between the axons, that this one fires, it makes some kind of regenerative potential here, maybe not a full spike, which can then propagate backwards, possibly block in this region, and you get a spikelet, or not block and you get a full spike. This was the hypothesis. And they tested this with gap junction blockers, which if you have a blocker in the median, sort of closes this junction or hypothetical junction, and you don't see the spikelet, or you see a reduced spikelet. Or he would, if he's recording from this cell, he was able to acidify the interior of this cell. And gap junction conductances are very sensitive to pH, although in different ways, depending on the channel. But he showed you could block the spikelet that way also, by manipulating the pH. And this is important also for what Rafa is doing, but important here. It connects in 36 channels, open with acidification, but connects in 45 channels, closed with acidification. So there's a functional difference. Okay, so let us assume that these junctions actually exist. This is not going to be established in stone until there's freeze fracture. But there's a lot of indirect evidence, and let's suppose that they actually exist. I still haven't answered the question, how do you get a collective ripple frequency oscillation out of a system like this? So we're assuming you have pyramidal cells like so, basal dendrites, apical dendrites, axons, a junction somewhere there. And we can estimate where this is from Sebastian Schuchmann's data, somewhere of the order of 100 to 200 microns from here. So I want to show you now a model in which you can take this kind of behavior and get a collective oscillation out of it. Now this was not straightforward because if the period of a ripple oscillation is about 5 milliseconds. But the time it takes for a spike to go from there to there, it's about 1/4 of a millisecond. So the problem, the mathematical problem is, where does this time constant come from? Where does the period come from? How can you account for the period? And it turns out you can account for the period of the ripple with topology. It's not a matter of the kinetics of membrane channels or synaptic currents. It's a matter of the topological structure of a network. And this sounds, I know, sounds like nonsense, but it works. In simulations, it works. So again, this is the assumption about 2 cells, and let's say we'll take a whole bunch of cells, maybe 500 or 1,000 or 10,000, whatever you like, and hook them up. So this is supposed to depict a random graph. So if you think of each of these numbers as being a cell or an axon, if you like, and so this is what's called a random graph. Now, the random graph is constructed so that each cell talks to, on the average, 1.5 to 2.5 other cells. You don't want too many, but you have to have more than one to be above what's called the percolation limit. And I also did simulations with trees like this. And the reason for that is random graphs like this have cycles in them, but a tree does not have cycles. So I wanted to show that you could get a collective oscillation even without cycles. So the assumptions that I made were very simple. First, you have this structure. Second, you have this property where a spike goes from one axon to another. And the third assumption was that there's some background noise. In other words, every now and then a spike would occur just by itself. An ectopic spike is called. So suppose you have an ectopic spike, which is pictured here with this brown square. So this is spontaneous action potential. And with this assumption of spread, it spreads, and then the next step it spreads some more, and eventually it kind of fills up the system, and everything becomes refractory and it stops. And then at some random time later, there's another ectopic spike. And what this produces is a sort of a series of waves that go through the system, they interfere with each other and die out. And as luck would have it, if you have not too much noise, not too little noise, a reasonable connectivity, which agrees with dye-coupling experiments, what comes out of this looks like a ripple. So the reason for the period is, even though this time is very short, the time to get a wave of excitation through this whole system is quite long because it's a whole series of steps. That's why you don't want to have too many gap junctions. So if you have not too many, not too few, you have this spread, it takes lots and lots of steps, and that's where you get the period. That's what I mean by topology determining the period rather than a membrane current kinetics. This is, for people in statistical physics, this is very easy to grasp, but with classical electrophysiology backgrounds, somehow it's maybe unfamiliar. So just to reiterate, there's nothing mysterious about this. If you think about the heart, the myocardial cells are coupled by gap junctions, and which allow a cardiac action potential to pass from one cell to another, which is a good thing, or we'd all be dead. And that doesn't take very long. But if you look at an EKG, so where this is 1 second, no, this is 0.2 seconds, 200 milliseconds. So this is P wave, so this is atrial contraction, and then there's some interval, and then you get the QRS complex. This interval is hundreds of milliseconds. It's the same idea because there are lots and lots of steps to go from the sinoatrial node through the whole ventricle. And these steps add up, and so you get these long time intervals. It's actually totally trivial, but different than what people are used to thinking about with gap junctions, say in the inferior olive, where you have oscillating cells, and what the gap junctions are doing is kind of coupling these oscillators together. This is different. So now a bit of relevance to clinical matters. Ripple-like events occur prior to seizures, at least with some kinds of brain pathology. And this is an example of that. So this is a recording from an epileptic patient with ECOG, or electrocorticography. So these are electrodes on the surface of the brain. This patient was being evaluated for epilepsy surgery. So there was a lesion on MRI imaging, but the surgeons wanted to know that the seizures in that patient actually were arising from that lesion and not from somewhere else or not from multiple places. So to do that, a grid of electrodes was placed on the brain, and many seizure events, so-called electrographic seizures, were recorded, and you could tell that, in this case, that they all came from more or less the same place. Now the thing is that when this kind of measurement is made with a high enough bandwidth, what you can see is this. So this is the seizure here. This is about 11 seconds of data. That's the seizure. But prior to that is a ripple that looks like this. So this is 150 hertz. So it's low amplitude compared to the seizure itself, but very high frequency. And the question is this abnormal phenomenon generated in the same way as the normal ripples? And I would claim that it is, although by a different population of cells, but the mechanisms are similar. So in order to test this idea, Miles Whittington replicated this kind of abnormal ripple phenomenon in a slice of, or in slices of neocortex. This is from rat. So this is data from a rat, and he's blocked chemical synapses, so sort of analogous to the original Dragoon experiments, and then he puffed on a solution at alkaline pH. And when he did that, he got a ripple oscillation. I mean, it's longer than a ripple. Looks very much like the human data. And when he recorded simultaneously the layer 5 pyramidal neuron, he saw this mixture of spikelets and action potentials, very much like what Andreas Dragoons had seen 20 years ago, only this time it was continuous. And here it is kind of blown up a bit. So a mixture of spikelets and action potentials correlated with a local field that's oscillating at 150 hertz. This is now a model of 3,000 and some model pyramidal neurons doing the same thing. And with this cell, it's subject to a driving current, which keeps the cell hyperpolarized and gradually depolarizes it. And you see here just spikelets, and then there you get this mixture that looks like the experiment. So the claim is that this phenomenon is ripple-like, it's produced by gap junctions, and may be induced in patients by alterations of pH in the brain. And this is something that needs to be tested in patients, first in slices and animals with the proper pH technology. So I'm talking to you. But I think this is something that really is going to matter. Let's skip through this. So just to repeat, I've tried to show you evidence that pyramidal cell axons are coupled together in a particular way, that coupling can lead to ripple frequency oscillations, that these oscillations could explain the data, the in vivo data on ripples, although I haven't shown all of that, but I will just give you two of the experiments. So as I said before, one prediction was that the action potentials during ripples should be antidromic. And that certainly looks to be the case here. This is in the experiment. This is a spike coming off a hyperpolarization. Orthodromic spikes don't do that. They come off of depolarizations. And Andreas Dragoon and colleagues did other measurements that very much suggest that these spikes are all antidromic during the ripple, which is very strange when you think about it. This is now a model based on this picture I showed before. And you see the same kind of thing. You can get a field oscillation, ripple frequency, during a hyperpolarization with antidromic spikes. And in the model, I know these are antidromic because I can look at what the axon is doing. So this is one prediction that at least in vitro is verified, very counterintuitive prediction. And makes you kind of wonder, what the hell? I mean, how is it that you have a normal phenomenon which is involved in memory consolidation and yet the action potentials are not arising because of synaptic integration, which is what we all think they should be doing. I mean, that's how the brain is supposed to work, as cells integrate their inputs and make decisions and send outputs to other cells and blah, blah, blah. And yet here, these spikes are arising in a way that just seems totally stupid. This is not the result of synaptic integration. It's something else. And I don't have an answer to that. I think this is a profound question. So this is the other prediction, was that an oscillation in the ensemble of axons will also produce ripple frequency excitatory synaptic currents in pyramidal cells. And in this paper by Nikolaus Meyer and colleagues, they observed that. They're small, but that's okay. They occur regularly. So now the question is, what does this mean? This is the normal phenomenon. This is a recording from human epileptic neocortex, the slice has been removed from the patient. Again, this is field potential, ripple frequencies, and evidence of antidromic activity with these spikelets. This is normal, or sort of an in vitro correlate of something normal. This is clearly abnormal. Why is this? So I'll leave you with a question, and these are some of my many colleagues. Thanks.
So I would have a question. Of course, we spoke about leaving outside mixed synapses for this talk and things like that. When you have an axodendritic synapse, electrical synapse, it seems that it does not participate in high frequency activity, but on low frequency activity.
Right. Yes, I think it's doing something different.
Okay, so that can also be predicted by what you have done with your models, or have you addressed that?
No, well, I don't know what the mixed or hippocampal mixed synapses are doing in this kind of context. I don't have a good idea. I don't know how to think about it. But certainly there's some high frequency correlate because you see spikelets. So it's not really DC coupling. I don't know. Have you ever seen a spike in the multi-axon produce a spike in the pyramidal cell?
No, we only reported the most spike itself, only the zone.
Okay. But certainly you see spikelets, which are, this is not DC coupling, at least not pure DC coupling. But I don't know how to take this kind of thing and put it in a context like this. That's why I didn't get into it, because I don't know how to do it. And the same thing in the spinal cord. Well, there are all these mixed synapses in the spinal cord. I don't have a good feel for what they're doing. The Mautner cell, I have something.
What I feel very interesting is that every lab I have visited in the whole world have seen spikelets, but they don't pay attention to them.
Right, exactly.
Everybody says that they are there.
Yeah. But Richard went further. He said it's an artifact. They're there, but they're not there. Yeah. So, but the gray area can be also. So what can I say?
Yes.
Hi. I have a question about the, they are there, in which moment do you think they participate? I mean, who triggers the opening, the gating? Because we do have like ripples all the time, but so if the signals don't participate once that is ongoing,
Yeah.
but maybe it's necessary for the beginning or not. I mean,
so they're right. So they're I can give a partial answer. So there are a couple of things. One is no one knows what initiates sharp waves. This is the last I heard. Is that in Berlin? No. There are some ideas. So no one knows. But the other thing is that gap junctions are modifiable by neuromodulators, particularly dopamine, but also serotonin and acetylcholine, and probably other things, and also by pH. So there are lots of factors in the brain that can alter their conductance quite significantly. And in fact, in the retina, there's an in vitro retina preparation. So you look at cells that are coupled, and people have done microscopy. So in one state, you see the gap junction looks like a snake, kind of like that. And then with dopamine, it doesn't look like this, it looks like this. So it actually changes the whole structure of it. And the thing is, the snake is conducting and the disc is not conducting. So I'm guessing that in the sharp wave state, it's determined by modulators. That's just a guess.
So the second question is, you think the couple between gap junction is in the whole pyramidal layer, in all cells, or maybe just in few? I mean, because maybe the beginning, or once that is ongoing, the ripple is not necessarily the synapse, but maybe the consequence is producing LTP or LTD because these high frequency events in the samples, in the pyramid.
Well, so, I mean, yeah, so this, the NAGI data, the best NAGI data is all CA1, and it's not, he doesn't look at the whole length of CA1, only a piece of it. But at least within that piece, this kind of stuff is pretty widely distributed. So I would guess at least there, it's kind of random, but more or less uniform, that's a guess. In cortex, we have only indirect evidence. So you can produce gamma oscillations in superficial layers of auditory cortex, and you see spikelets, and you block the oscillations with carboxylol. So we think superficial regular spiking pyramidal cells are coupled this way. And there's another experimental model where if you look at deep regular spiking pyramidal cells, you also see spikelets. So we think they're coupled. And then there is this picture I showed with, this is intrinsic bursting cells. They appear to be coupled, at least in frontal cortex. So I think it's widespread, but I don't know if it's uniform, but it's widespread. I don't know if that answers your question.
And you think produce LTD, the consequence of this?
So there's one paper about that, which is not specifically about gap junctions, but this, I can't remember her name, I think it was Bucalo, or Buffalo, it was either Buffalo or Bucalo. So she stimulated axons of pyramidal cells during some sharp wave-like state, and she showed that resulted in long-term depression for the inputs from CA3. You probably know this. So that's the only experiment I know that's relevant. And I don't know why that occurs, but that's what she found.
¿Alguien más?
In the tree-like graph model that you presented,
Yeah.
do you avoid cyclic graphs on principle, or because you explicitly want to avoid feedback loops, or because?
Yes. No, I mean, I don't think that the actual brain has any tree-like structures like that.
Yeah.
It was just sort of a mathematical thing to do, because this is a tree. I know it has no cycles, but it can still produce an
So
I shouldn't call it an oscillation. The power spectrum is not a delta function, but it's kind of like that.
Yeah. So it simplifies the model.
It
and
simplifies
it still
it.
gives the
That's right.
and the complexity of the graphs that you are using. How many nodes, how many vertices.
So I used a tree with 1,000 and some
something.
for instance.
And all vertices represent the same coupling.
Yes.
Yeah.
And then I used the random graph in that paper, I think, was 3,000 and something. But by the way, if you look at much larger systems, you start to see different phenomena that I didn't talk about. And also, it's also true that in random graphs, because there are cycles, there are conditions in which you do get re-entry. And when you can guess that there's re-entry because you kind of get this sort of irregular oscillation-like stuff, and then it goes bing, 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 it's perfectly irregular, and then stops. But it's very hard to prove that it's re-entry, because that would involve observing all of the cells. I don't know how to do that.
So it just occurs to me that maybe you can do random graphs with the data that we have already, with this big cloud of cells.
Yeah.
Maybe you can get from that this kind of distribution.
Yeah, I mean, you see lots of cells, but you don't know the connectivity.
Yeah.
So I hope I didn't confuse everybody.
I have a question of ignorance, a more basic question about the modeling. So you use a lot of knowledge and published information to generate a model, mathematical model, and then
What do you mean like this?
and then you use the model to confirm things that have already been published and then to make some predictions.
You mean how do you make something like that?
Yes.
Yeah.
Yeah. How do you use the model?
Yeah. So I started doing this in the 1970s by just accumulating as much biophysical information as I could and trying to get. This is not representing the anatomy exactly, but you get the area of the dendrites and the length of the dendrites and the area of the soma, and there are measurements of the input resistance, and you can estimate the capacitance, and then people do single channel recordings of sodium channels, and they publish kinetics of sodium channels and potassium channels and so on. So there's a lot of published data, but so it's not just made up totally. But on the other hand, a lot of it is made up because there is not. If you just depend on published data, there's no such thing as a precise biophysical model of a neuron. It's just a fantasy. So it's like I do the best I can, and I do lots of comparisons with recordings from different places, and it sort of works.
And then you validate your model by checking if things that are already published, they happen with your model.
Yeah, or things that experiments that are predicted. So like this was a while ago, like one prediction was make a model of a CA3 cell, and it's excitable in a certain way, and it can make dendritic calcium spikes. And the prediction was that if you stimulate it antidermically with inhibition blocked, you should get a burst of action potentials, which is not something that had been observed. And then Miles and John Jeffries did that experiment, and it was true. But on the other hand, the burst didn't look exactly like the model burst. I mean, it burst, but it's sort of a little different. So I don't know if I'm conveying the flavor. It's really an approximation, and it's the best I know how to do it. Now, the other point is that if there are inaccuracies in the biophysics of the channels, I don't think it changes these more general ideas about how the collective phenomena come about. Because that depends on a very simple idea, namely a spike goes from here to here. And that can be replicated with a cellular autonomy, a very, very simple abstract model.
Mas, I have a question. Ripples have been seen in other parts of the campus formation, like CA3, and it seems that this phenomenon can go to the cerebellum and coronary cortex. Do you think that this mechanism is similar, that the gap junctions that are in all the campus formation are different? But
I don't know. It's possible that, say, a ripple in CA1 could synaptically induce a ripple in the cerebellum, or it could be that if you isolate the cerebellum, it can make ripples on its own. And these are sort of experimental questions. that in principle are easy to do. I don't know the answer.
Okay, and the other question that I have is that if this mechanism is real, this phenomenon will be less correlated with the memory long-term potentiation theory, right? Because
No, I don't think so. I don't think there's any contradiction. At least I hope that's true. I mean, I don't want to claim that it's all gap junctions. That's certainly not true. I just want to say some part of the underlying mechanism is gap junctions. Then there's all the synaptic stuff on top of it. It's like with the developing nervous system, you look at developing spinal cord, this original circuits, the chemical synapses are not there. It's just gap junctions. So you see these kind of rhythmic oscillations. And then with development, the synapses start to form and more complicated things happen and it gets richer and richer. So that's the metaphor I have. There's this underlying something or other, which is electrical in the superstructure. Are you happy with that?
Although we have seen in all tissue that we have an immense amount of connexin, and it's incredible that it is not active all the time, right? So the conductivity is not there.
Yeah.
Doctor, you have been associating these spikes with the system with epilepsy, if I'm correct. I'm not an expert on encephalograms, so I don't understand so much about it. But you talked that at the beginning of every seizure, you cannot ascertain these type of spikes that appear?
No, this is only true for focal seizures, and I don't know that it's true for all focal seizures. What I can say is that we had this series. I was a minor collaborator. A series of patients from London with whose children with cortical dysplasias. There are 13 kids. Every one of those children, every seizure began with rippling. But that doesn't mean that a brain tumor, the seizures both begin with ripples. We don't know that.
I have another question. Some types of seizures had an aura before they start. So this could explain maybe the presence or not of an aura in the seizure.
No, I don't think so, because the aura corresponds to actually electrographic seizure. So but by the time the aura occurs, the seizure's already started. Even though it's subjective, usually it's subjective. Electrically, the seizure's already begun. So this is coming before that.
¿Alguien más? Bueno, pues muchas gracias. Y mañana va a dar una conferencia más general, menos específica del sistema nervioso, que creo que va a ser muy, muy interesante para todos. Así que los espero mañana. Thank you.